I saw an old car sitting in a field the other day, one of those beautiful, classic models from the 60s. You could still see traces of its original deep cherry red paint, but most of it was gone. Instead, there were these ugly brownish-orange patches bubbling up through the metal. We all know what it is. It's rust. And we understand rust as a kind of slow death. It's a chemical reaction called oxidation, where oxygen in the air relentlessly pulls electrons away from iron, causing it to decay and crumble into dust. It's the inevitable destructive end for a piece of strong, solid steel. But here's the thought that made me stop. That very same chemical reaction, the one that's turning that car into a pile of reddish dust, is happening inside your body right now, in every one of your cells. The only difference is we don't call it rust, we call it breathing. The same process that destroys metal is the very thing that powers your every thought and heartbeat. So how can the fundamental force of decay also be the fundamental force of life? And if we are, in a sense, just slowly rusting from the inside out, what does that really mean about the air we can't live without? That's the profound paradox we're diving into today. This deep dive is all about unpacking that exact contradiction, how the very air that sustains us also initiates a slow, silent decay within our bodies. We're going to journey back to Earth's ancient past, exploring a time when oxygen wasn't life-giving, but a catastrophic poison that reshaped life on a planetary scale. And then we'll leap to the cutting edge of science, contemplating a future where humanity might not even need to breathe, where we could engineer ourselves beyond oxygen's embrace. It's a truly wild ride, from the microscopic battle within your cells to the grand tapestry of Earth's evolution and beyond. So let's begin with the deeply personal impact of this paradox. We're going to investigate the intricate biochemical pathways and, the cellular defense mechanisms that constantly work to mitigate the continuous oxidative stress induced by essential oxygen metabolism, and what their implications are for human aging and the development of chronic diseases.
Yeah, and I came across a story that really brought home the silent, lifelong battle our bodies fight. It was just quite powerful. It was about an elderly man in a small coastal town in Japan, a man named Haru. He'd spent 70 years as a kokeshi doll maker. His hands, which had once carved smiles onto wood with surgical precision, now trembled ever so slightly. He told his grandson it felt as if a slow, patient rust was settling into his joints, the same way the sea air corroded the iron hinges on his workshop door. He'd sit by the window, taking deep, steady breaths of the ocean air that he loved, the very air that had fueled his life's work. It's a powerful image to think about. The same oxygen that powered the muscles in his hands for decades was now, through the slow burn of oxidative stress, contributing to the tremors that were forcing him to put his tools down. For Haru, oxygen was both the energy for his art and the imperceptible thief of it. As a science communicator, this story makes me see the data on cellular aging, not as abstract figures, but as the lived experience of people like Haru. We talk about mitigating oxidative stress, but what we're really talking about is giving people more time with the things they love, trying to hold back that inevitable patient rust, one breath at a time.
Yeah.
It just underscores the profound link between our daily choices and this intricate internal war.
Haru's experience really paints a vivid picture of this slow burn. So what exactly is that slow burn happening in our bodies? You see, oxygen is truly a profound paradox, almost like a biological double-edged sword. On one side, it's absolutely non-negotiable for life. It's the critical ingredient for cellular respiration, the fundamental process that extracts energy from the food we eat, powering every single one of your cells, from the blink of an eye to the beat of your heart.
Can't live without it.
Exactly. Without it, everything stops. It's our invisible lifeline, our essential fuel. But on the other side, oxygen is incredibly reactive. Its very nature, its insatiable desire for electrons, means it's constantly stealing them from other molecules within your cells.
Like tiny thieves.
Think of it like a tiny, invisible pickpocket.
Yeah.
Disrupting the delicate molecular bonds that hold everything together, leaving behind what you could call cellular toxic waste. This electron theft leads to the creation of highly unstable molecules known as reactive oxygen species, or ROS, and reactive nitrogen species, RNS.
Right, ROS and RNS.
These are not abstract concepts. They are actual aggressive molecules that your body naturally forms during normal cellular metabolic processes, especially within your mitochondria, those powerhouses of your cells.
The energy factories.
It's a cruel irony that even our internal energy generators, the mitochondria, aren't perfectly efficient. They actually leak electrons during energy production, inadvertently creating more of these damaging ROF.
A bit leaky.
You can imagine these ROS and RNS as microscopic bullets, constantly firing and causing collateral damage to your vital cellular components, your precious DNA, the proteins that make up your enzymes and structures, and the very membranes that form the protective walls of your cells. It's a constant internal assault. When your body's production of these pro-oxidants, these molecular muggers like ROS, RNS, and even reactive chlorine species, overwhelms your natural antioxidant defenses, that's what we call oxidative stress.
Imbalance.
Exactly. It's an imbalance, a battle where the bad guys start to outnumber the good guys. Luckily, your body isn't defenseless. It has an incredibly sophisticated built-in system of defense. You have enzymatic antioxidants, like specialized cleanup crews, such as superoxide dismutase, often called SOD, glutathione peroxidase, or GPX, and catalase. These enzymes are like highly trained special forces, designed to quickly sequester and neutralize ROS and RNS, preventing them from causing widespread damage.
Your internal cleanup crew.
Right. And beyond these enzymes, your body also produces non-enzymatic antioxidants internally, including powerful molecules like glutathione, uric acid, and even melatonin, along with various metal-binding proteins and bilirubin. But this internal fight against cellular rust isn't one your body fights alone. Your diet plays a huge supportive role in bolstering these defenses.
Absolutely crucial.
That's where dietary antioxidants come in. We're talking about compounds broadly known as polyphenols, which are abundant in the vibrant colors of fruits, vegetables, and even things like green tea.
Eat the rainbow, as they say.
Exactly. Think of all those blues, reds, purples, and berries, greens, and leafy vegetables. They're packed with these protective compounds. And then there are the antioxidant vitamins, like vitamin C, vitamin E, and vitamin A, along with beta-carotenes and essential minerals like selenium, manganese, zinc, copper, and iron. Take a chedin, for example. It's a natural flavonoid you find in everyday foods like parsley, onions, oranges, and even the calming chamomile tea. Research has shown it has remarkable protective effects for your heart, your brain, and your kidneys. It's also proving beneficial in managing metabolic syndrome. But here's the catch. Getting enough of these beneficial compounds into your system effectively can be tricky because of something called bioavailability, how much your body can actually absorb and use.
Right. It's not just what you eat, but what gets in.
Precisely. That's why cutting-edge research is looking into novel delivery techniques like nanodelivery, using tiny particles to get these compounds where they need to go more efficiently.
So we've talked about this constant microscopic assault. The vital question then becomes, what does this silent assault mean for our long-term health? Well, the scientific data is overwhelmingly clear. Oxidative stress is directly connected to the onset and progression of a wide array of major chronic diseases.
Like a common thread.
Exactly. It's like a common thread connecting so many of the chronic health challenges we face, essentially accelerating the body's natural wear and tear. We see its fingerprints all over conditions like diabetes, where high blood sugar levels induce reactive oxygen species that damage the insulin-producing cells in the pancreas.
The B cells, right?
The B cells, yes. In obesity, increased oxidative stress contributes to the development of metabolic syndrome and non-alcoholic fatty liver disease, often called NAFLD. It's also deeply implicated in the progression of neurodegenerative diseases affecting millions, such as Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, ALS, and Huntington's disease.
Wow.
In fact, you'll sometimes hear Alzheimer's disease referred to as diabetes 3 because it shares common biomarkers with type 2 diabetes, like elevated levels of advanced glycation end products, or AGs.
AGs, okay.
And a nasty molecule called 4-hydroxytrans-2-nonanol, or HNE.
HNE, got it.
Beyond that, oxidative stress plays a significant role in cardiovascular diseases, contributing to the dysfunction of blood vessels and the hardening of arteries. And critically, it's involved in the initiation and progression of various cancers by causing damage to our DNA, leading to mutations and uncontrolled cell growth, including pancreatic cancers.
So it really touches almost everything.
Pretty much. The list goes on. Thyroid diseases, allergic diseases, pulmonary fibrosis, and even the progression of osteoarthritis in our joints. It's pervasive. Let's delve a bit deeper into some of these particularly damaging molecules. Advanced glycation end products, or AGEs, are especially insidious.
Okay, the AGEs again.
Imagine them as a kind of molecular superglue. They are deleterious structural modifications of proteins and even our genetic material, nucleic acids. They form through non-enzymatic reactions where sugars in our body spontaneously react with protein.
Some randomly.
Well, not enzymatically, it just happens. And here's the kicker. Oxidative stress significantly speeds up their formation through what are called glycoxidation reactions.
So the rust speeds up the glowing.
You got it. You find relatively high levels of AGs ubiquitously present in conditions like Alzheimer's disease, diabetes, and obesity, and their accumulation often directly correlates with the severity of the disease. Specific types like pentosiodine can form intermolecular crosslinks, which means they create unwanted rigid connections between proteins, stiffening tissues. CML is another common protein modification, where the protein structure is altered, often leading to loss of function.
So they really mess things up structurally.
They do. These are just a couple of examples of how AGs act like sticky glue, forming unwanted connections that stiffen tissues and disrupt normal cellular function throughout your body. What's truly concerning about AGs is their role in a vicious cycle. When AGs bind to their specific receptors for AGs, known as RAGE,
receptors, okay.
They don't just sit there inertly. Instead, they activate a cascade of damaging signaling events. This triggers the production of inflammatory cytokines like interleukin-1, interleukin-6, and tumor necrosis factor I.
Yeah.
Essentially, they throw gasoline on the fire of inflammation.
Inflammation goes up.
Right. These inflammatory responses, in turn, further exacerbate oxidative stress, creating a self-perpetuating destructive feedback loop.
So it just gets worse and worse.
Exactly. This age-rage interaction is deeply involved in the onset of pancreatic cancers, especially under hyperglycemic conditions, and it plays a causative role in diabetic kidney disease and the dangerous progression of aortic aneurysms. Then there's the damage from lipid peroxidation and direct DNA damage.
Okay, hitting the fats and the genes.
Precisely. When highly reactive hydroxyl radicals, those molecular muggers we talked about, interact with the delicate lipids in our cell membranes, they create harmful products like HNE and malondialdehyde, or MDA.
HNE and MDA.
HNE, for instance, is highly cytotoxic, meaning it's toxic to cells. It can bind to our nucleic acid bases, leading to DNA modifications that cause mutations and even contribute to cancer. It can also modify proteins, resulting in the loss of their function.
So it's bad news all around.
Definitely. HNE is actually used by scientists as a biomarker for disease progression and is implicated in conditions like osteoarthritis. Similarly, MDA, another product of lipid peroxidation, forms adducts with our genetic material that, without proper DNA repair mechanisms, can also lead to mutagenicity and carcinogenicity. And ROS also damages DNA directly. Yes, ROS directly damages our DNA by oxidizing one of our DNA building blocks, 2-odeoxyguanosine, into 8-hydroxy-2-odeoxyguanosine, or 8-oxo-DG.
8-oxo-DG.
Which serves as a widely used marker for oxidative stress and DNA damage, almost like a cellular smoke alarm.
Oh, okay.
Interestingly, some modern cancer drugs, like diosgenin, strategically leverage this by promoting DNA damage in cancer cells via ROS, forcing those cancerous cells to self-destruct.
Turning the weapon against the enemy.
Sort of, yeah.
Yeah.
Finally, we have nitrative stress caused by reactive nitrogen species, or RNS.
RNS.
The key players here are nitric oxide, or NO, and peroxynitrous acid, NOH. Now, nitric oxide is actually a crucial signaling molecule in our body under normal conditions.
Right, I've heard of NO.
It's involved in everything from blood vessel dilation to nerve signaling. But when produced in excessive amounts, often by overactivation of certain enzymes, it can become problematic. Too much of a good thing.
Exactly. When NO reacts with the superoxide radical, it forms peroxynitrous acid, a highly reactive and damaging species.
Oh no, H. Sounds nasty.
It is. Peroxynitrous acid is particularly problematic because it causes tyrosine nitration, also known as nitrative stress, and S-nitrosylation of proteins, modifying proteins again.
Right. These chemical modifications can significantly alter how proteins function, contributing to a range of neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's disease. What's more, peroxynitrous acid can also generate those highly reactive hydroxyl radicals we just talked about, further increasing the overall oxidative stress in the cell, creating a cycle of escalating damage.
So it feeds back into the ROS problem too.
Precisely. It's all interconnected.
So given the widespread and insidious nature of this damage you've described, the immediate question that springs to mind is, are we simply at the mercy of this slow burn, or can we actually intervene? The good news is there are fascinating therapeutic frontiers being explored, constantly pushing back against this cellular rust. One area of intense interest is targeting those advanced glycation end products, the AGEs.
I gotta stop the glue.
Exactly. Researchers have investigated compounds designed to act as AG inhibitors and even AG breakers. For example, compounds like aligebrium, sometimes called ALT-711, were developed with the ambitious goal of actually breaking apart preformed AG protein crosslinks, essentially, trying to untie that molecular superglue.
Did it work?
Well, another compound, aminoguanidine, or pimidine, showed promise as an anti-glycating agent, aiming to prevent AGEs from forming in the first place. However, these compounds have had limited clinical success.
Why is that?
It turns out that the sheer complexity of AG structures and their intricate formation mechanisms make them incredibly difficult targets to hit selectively and effectively within the body. Almost like trying to untangle a massively knotted ball of yarn with a single thread.
Complex problem.
More recently, there's been growing excitement around RAGE inhibitors. Remember, RAGE is the receptor that AGs bind to.
Right, the doorway.
Triggering that cascade of inflammation and oxidative stress. These inhibitors work by essentially blocking the doorway. They bind to the RAGE receptor, preventing AGs and other harmful ligands like the amyloid beta peptide implicated in Alzheimer's from binding and initiating that destructive feedback loop.
So blocking the signal.
Exactly. A promising example is a compound called FPSCM1. In lab studies, it has shown neuroprotective effects, suppressing the influx of circulating amyloid beta into the brain and improving cognitive performance in mouse models of Alzheimer's disease.
That sounds promising.
It does. Another RAGE inhibitor, azelargon, is actually in clinical trials for both Alzheimer's disease and cancer, which is a huge step.
Clinical trials, wow.
Yeah. The challenge, though, is that RAGE is like a master keyhole, capable of binding to many different damaging molecules, making it difficult to achieve highly selective inhibition without unintended consequences elsewhere in the body.
Right, the selectivity issue.
Beyond these specific compounds, novel approaches are rapidly emerging. We're seeing the development of single-atom nanozymes.
Nanozymes.
Yeah, which are incredibly tiny, engineered catalytic materials designed to attenuate oxidative stress by efficiently sequestering both reactive oxygen species and reactive nitrogen species, almost like hyper-efficient molecular sponges.
Tiny sponges, cool.
And, circling back to what we consume, dietary polyphenols, those vibrant plant compounds we talked about, are not just general antioxidants. They can specifically regulate the age-RAGE axis, helping to calm that inflammatory feedback loop.
Targeted action.
And they can even positively influence the gut-brain axis, potentially playing a role in preventing neurodegenerative diseases. It's a truly multi-pronged approach. This intricate, lifelong battle against cellular decay, this slow rust in our bodies, brings up a fascinating paradox we observe in the animal kingdom. The elephant paradox.
Yes, Peto's paradox.
Exactly. Think about it. Massive animals like elephants and whales, which live for decades, rarely get cancer despite having billions more cells than we do. Why?
More chances for things to go wrong, you'd think.
Right. It's because they possess multiple copies of the TP53 gene, which is often called the guardian of the genome.
The tumor suppressor gene.
Precisely. This gene is absolutely critical because it acts like a vigilant sentinel, detecting damaged cells very early and then ordering their programmed death, preventing them from turning cancerous. Humans, on the other hand, have only one copy of this crucial gene.
Just one copy.
This highlights the incredibly delicate balance required for genetic therapies. Too much TP53, and your body might aggressively kill healthy cells, potentially accelerating aging. Too little, and cancer gains an advantage. It's a constant tightrope walk between protecting ourselves and risking unintended consequences.
A very fine balance.
We've explored oxygen's role in our personal health, from the silent cellular rust to cutting-edge therapies that try to fight it. But its story on Earth is even more dramatic, a tale of monumental transformation that redefined life itself. Now let's analyze the profound evolutionary compromises and adaptive pressures that arose from the Great Oxidation Event, fundamentally shaping Earth's biodiversity and informing our entire astrobiological search for diverse life forms beyond oxygen-dependent metabolisms.
Yeah, this is where things get really planetary in scale. And again, a story helps illustrate it. I was reading the field notes of an astrobiologist, Dr. Eris Thorne, who was working in the acidic, sulfur-rich caves of Villaluz in Mexico.
Hostile environment.
Utterly hostile to us, yeah. A world without light, where the air itself is toxic. Yet it's teeming with life bacterial colonies called snow-tites that drip from the ceilings, getting their energy not from oxygen, but from hydrogen sulfide.
Snow-tites.
Oh.
Charming.
Yeah. Eris wrote about a moment of profound clarity he had in that toxic darkness. Surrounded by life that would be poisoned by the air he breathed, he imagined the great oxidation event not as a dawn, but as a holocaust.
A holocaust, wow.
He envisioned an ancient Earth, rich with anaerobic life, suddenly choking on a new corrosive gas oxygen. What we call the beginning of our world was the end of theirs. He said that every sample he collected felt like finding a ghost, a survivor from a lost world.
That's a powerful image.
It is. His search for alien life wasn't just about looking to the stars. It was about understanding the profound evolutionary compromise that allowed us to exist, a compromise built on the extinction of countless other possibilities. Reading that, I had to stop and just think. We are so conditioned to see oxygen as a universal symbol of life. But Eris's experience suggests that on a cosmic scale, it might be a planetary signature of a massive extinction event.
Whoa.
It completely reframes our search for life beyond Earth. We might be looking for our biological cousins when we should be searching for the ghosts of a different, more ancient lineage that our own ancestors defeated.
That's incredible. Eris's perspective really shifts our entire understanding of oxygen's history on our planet. It makes you rethink everything, doesn't it?
Absolutely.
So let's unpack this monumental shift known as the Great Oxidation Event, or GOE. Imagine Earth around 2.4 billion years ago. It was an alien landscape. The atmosphere and oceans were vastly different from today's blue skies and clear waters.
Totally different planet.
It was a world ruled by anaerobic microorganisms, life forms that absolutely thrived in low-oxygen environments. The oceans were likely murky, rich with dissolved methane, sulfur compounds, and carbon dioxide. The sky was probably a hazy, rusty orange, and the seas had a distinct greenish hue.
Orange sky, green seas. Wild.
Oxygen, in anything like the concentrations we know today, was nearly absent. Then came the GOE, a truly catastrophic biochemical revolution that forever altered our planet. It all began with tiny single-celled organisms called cyanobacteria.
Blue-green algae.
Exactly. These incredible microbes evolved the ability to perform oxygenic photosynthesis, a revolutionary trick that used sunlight and water to create sugar for their energy, and crucially released oxygen as a byproduct.
A waste product initially.
A waste product that changed everything. As these cyanobacteria flourished and multiplied, they began to slowly but relentlessly pump oxygen into the oceans, and then gradually into the air. And what happened? A massive planet-wide extinction event. For most of the life forms existing at that time, anaerobic life that had evolved in an oxygen-free world, this new gas was pure poison. It was, as Eris put it, a holocaust for them.
The first great pollution event, really.
Yet, amidst this planetary catastrophe, a painful and profound compromise emerged. A few tenacious organisms, through random mutation, developed the capacity to not just tolerate oxygen, but to actually depend on it, using it as a far more powerful fuel source for their metabolism.
Adapt or die.
Pretty much. This wasn't an immediate triumph. It was the reluctant beginning of life's addiction to oxygen. It forced biology to fundamentally mold itself around this highly reactive, dangerous molecule just to survive and unlock immense energy. This marked a profound trade-off. A new, incredible source of power for life, but at the inherent cost of constant cellular decay, a continuous struggle for longevity and safety in the face of rapid energy production.
Power versus longevity.
Exactly. Scientists pinpoint the GOE to approximately 2.4 to 2.3 billion years ago. We trace its timing through various biological markers preserved in ancient rocks and by examining minerals formed in the presence of oxygen. For example, sulfur isotope studies indicate that oxygen eventually reached a steady, albeit still low, atmospheric level, roughly 0.001% of current levels, and the ozone layer was established, making oxygen a permanent, albeit initially sparse, component of our atmosphere from that point forward.
A tiny fraction, but enough to change everything.
One of the most compelling geologic witnesses to this ancient transformation are banded iron formations, or BIFs.
Ah, the BIFs, Earth's rusty diary.
That's a great way to put it. These are truly Earth's fossilized diary, distinct units of sedimentary rock found almost exclusively from the Precambrian age, some of the Earth's oldest, dating back as far as 3.7 to 3.8 billion years ago. They can be incredibly vast, up to several hundred meters thick and extending for hundreds of kilometers, and they consist of repeated, thin layers of iron oxides like magnetite and hematite, alternating with iron-poor chert.
Like geological pinstripes.
It's like nature's own layered cake. But instead of frosting, it's ancient rust. The prevailing theory for their formation is utterly fascinating. Oxygen, produced by those photosynthetic cyanobacteria, began combining with the vast amounts of dissolved ferrous iron, Fe2+, that saturated the ancient oceans.
Iron just floating around in the water.
Loads of it. This reaction formed insoluble ferric iron oxides, Fe3+, which then precipitated out of the water and settled onto the ocean floor. The distinct bands we see are thought to result from cyclic variations in oxygen production, perhaps driven by seasonal algal blooms, much like tree rings or annual layers of sediment.
Blues and busts of oxygen.
Exactly. These formations are not just geological curiosities. They hold immense economic importance even today. BIFs account for more than 60% of global iron reserves and provide most of the iron ore mined worldwide.
Our cars, our buildings. A lot comes from ancient rust.
Absolutely. You'll find these immense deposits in places like the Hamersley Range in Australia, the Iron Range in the U.S., Itabarite in Brazil, and Achan in China. For these massive BIFs to form, several crucial preconditions had to be met. The deep ocean waters had to be incredibly rich in soluble iron, meaning they were anoxic or oxygen-free to allow the iron to remain dissolved and transportable.
No oxygen to rust it out immediately.
Right. And critically, these waters could not be euphsinic, meaning they couldn't be rich in hydrogen sulfide, because that would have caused the ferrous iron to precipitate as pyrite, a different mineral.
So anoxic but not sulfidic. A specific window.
A specific window, yes. And of course, an oxidation mechanism, the nascent oxygen from cyanobacteria, was required to convert that soluble ferrous iron to insoluble ferric iron, allowing it to precipitate out. The peak of major BIF deposition coincides precisely with the permanent appearance of oxygen between 2.41 and 2.35 billion years ago.
Good timing fits perfectly.
The end of major BIF deposition around 1.85 billion years ago is largely attributed to the widespread oxidation of the deep ocean, following what's called the Holland Ocean model, where the ocean's depths finally became oxygenated. Interestingly, the massive Sudbury Basin impact, which occurred 1.849 billion years ago,
Big asteroid hit.
huge one, is even hypothesized to have caused significant ocean mixing and deep ocean oxygenation, potentially playing a role in ending this era of BIF deposition.
Nature's giant spoon stirring things up.
We also find a smaller number of later BIFs from the Neoproterozoic era, between 750 and 580 million years ago.
Much later.
Yeah. These are often associated with severe planet-wide ice ages, famously referred to as snowball Earth events.
When the whole planet froze over, maybe?
Possibly. During these extreme glaciations, the oceans might have become largely anoxic again, allowing iron to accumulate, and then precipitated when the seas reoxygenated after the planet thawed.
Freeze, collect iron, thaw, rust, repeat.
Something like that. An alternative theory suggests these later BIFs formed from metal-rich brines near active rift zones, hinting at the complex geological dance happening beneath the ice.
If we connect all this to the bigger picture, the Great Oxidation event wasn't just about oxygen appearing, it was a complete planetary makeover, leading to cascading, irreversible changes across all of Earth's spheres, land, sea, and air. In the biosphere, the evolution of oxygenic photosynthesis by cyanobacteria led to an enormous increase in primary production. Imagine ecosystems becoming at least 10 times more efficient at generating biomass than other types of metabolism.
A massive boost in life's productivity.
Huge. While this massive oxygen output undeniably caused a mass extinction for existing anaerobic life, it simultaneously created an incredibly strong evolutionary pressure for aerobic respiration, the oxygen-using metabolism.
Right.
This new form of respiration, which utilizes oxygen, releases significantly more chemical energy in the form of ATP than any anaerobic pathways. This higher energy yield was an absolute game changer. It was like upgrading life's metabolic engine from a sputtering bicycle to a high-performance race car.
Fuel injection for life.
Pretty much. This abundance of energy facilitated the evolution of all multicellular and complex life as we know it, making possible everything from tiny insects to giant whales. Genomic analysis even suggests that aerobic respiration arose remarkably quickly in cyanobacteria after they began producing oxygen, a testament to its powerful evolutionary advantage.
They adapted fast.
The geosphere also underwent irreversible changes. The oxygenation led to the widespread formation of red beds, which are iron oxide-containing sedimentary rocks, turning vast swaths of land and shallow waters a rusty red, essentially a global sign of planetary oxidation. Earth literally rusted.
In a way, yes. We also saw the formation of widespread iron oxide soils and massive gypsum deposits. Conversely, minerals that readily decompose in the presence of oxygen, such as pyrite, uranite, and siderite, became exceedingly rare in rock formations after 2.4 billion years ago, providing tangible geological evidence of this atmospheric shift. The rocks tell the story. They do. In the atmosphere, the gradual accumulation of oxygen directly led to the formation of the protective ozone layer in the stratosphere, shielding the planet from harmful UV radiation, which was critical for life to eventually colonize land.
The ozone layer, crucial.
Absolutely. Additionally, the increased photosynthesis by cyanobacteria led to a significant decrease in atmospheric carbon dioxide, impacting the global climate. Climate change, ancient style.
Right.
And finally, the hydrosphere mirrored these changes, with increased oxygen and decreased carbon dioxide, which sped up the final depletion of soluble ferrous iron in the oceans, effectively ending the era of massive BIF deposition. It's also important to note that the GOE profoundly coincided with a major tectonic transition around 2.5 billion years ago. Plate tectonics were changing too.
Big time. Continent formation increased dramatically, and continental crust began to rise significantly above the oceans. Land volcanoes became dominant, erupting oxidized gases like sulfur dioxide, instead of the hydrogen and hydrogen sulfide that characterized earlier volcanic activity. Even the volcanoes changed their breath. Furthermore, the very form of plate tectonics dramatically shifted from what called a stagnant lid crust system, where the Earth's crust was mostly a single, slowly overturning piece, to active subduction, where cooler, stiffer ocean lithosphere began to plunge into the mantle, driving dynamic plate movements.
The conveyor belt started moving.
Exactly. This advent of active subduction fundamentally changed geochemical cycles on Earth, and may have been an important feedback mechanism, potentially influencing both cyanobacterial evolution and the GOE itself. It was a complete planetary system transformation.
This profound evolutionary story makes you wonder, doesn't it? If oxygen was such a violent catalyst for complexity on Earth, a force that dramatically pruned one form of life to allow another to flourish, is complex life elsewhere in the universe always dependent on it, or something strikingly similar?
The big question for astrobiology.
Right. Astrobiologists are constantly grappling with this question. Is complex alien life possible without oxygen? And increasingly, they're moving beyond oxygen as the sole biosignature they look for, because, as we've discussed, oxygen can be produced abiotically, meaning without life.
False positives are possible.
Exactly. Plus, life on Earth existed for hundreds of millions of years without detectable atmospheric oxygen, so assuming all life must have it is a very Earth-centric view. So researchers are now looking for a broader array of alternative biosignatures. This includes seeking oxygen in conjunction with strongly reactive species like methane. Finding both together would strongly imply a continuous biological source for both, as they would otherwise react and disappear.
Gases that shouldn't coexist without life constantly making them.
Right. Or they look for other gases like nitrous oxide, which could indicate specific metabolic processes like partial denitrification. They're also considering more agnostic indicators, which don't depend on a specific biochemistry. Like what? This could be subtle clues like isotope ratios. Lighter isotopes tend to react more efficiently, and a significant abundance of them in certain compounds could indicate biological processing.
Life prefers the lighter stuff sometimes.
Or even atmospheric complexity itself, or seasonality in atmospheric composition, like changes in gas levels that wax and wane with the planet's seasons, suggesting an active biology.
Planetary breathing, in a way.
But the core of the argument for oxygen's necessity for complex life often comes down to its incredible energy yield.
It's just so powerful.
Oxygen simply offers significant advantages over other potential electron acceptors. It boasts the highest redox potential, which means it can grab electrons with immense force, leading to a much greater energy yield, or ATP, compared to alternatives like sulfate, nitrate, or ferric iron.
More bang for your buck, metabolically speaking.
Exactly. This high energy yield means organisms can produce vastly more ATP, the cellular energy currency, far more efficiently. Secondly, the reduction of oxygen to water proceeds much more rapidly compared to, say, the sluggish reduction of sulfate to sulfide. Oxygen is a highly efficient electron acceptor, facilitating much faster reaction rates.
Speed matters, too.
It does, especially for organisms with high metabolic demands, like anything with muscle movement or complex nervous systems. These factors combined mean that the use of oxygen as an electron acceptor provides a substantial evolutionary advantage. It literally provides the fuel and the speed to support more complex and energy-demanding cellular processes, thereby facilitating the evolution of diverse multicellular organisms and ultimately intelligent life. So, could alternative metabolisms support complexity?
That's the million-dollar question.
While sulfur-based metabolisms exist, the limits to the complexity they can support in the absence of oxygen are largely unknown. Did life on Earth simply reach that metabolic limit before oxygenic photosynthesis evolved?
Maybe sulfur life just couldn't get big or fast enough.
There's also the intriguing idea of hydrogen photosynthesis or respiration, which could hypothetically produce a respectable plus 0.63 volts of energy, not as high as oxygen, but a significant amount that seems close enough to potentially support a complex organism.
A decent alternative, perhaps.
However, hydrogen isn't a good biosignature for detection from afar, as it's common in many abiotic processes. What about even stronger oxidizers like fluorine?
Whoa, fluorine.
Yeah. While fluorine certainly has a higher redox potential than oxygen, it's generally considered unsuitable for biological systems. Fluorine reacts violently with almost all organic molecules, effectively destroying biological tissues before any controlled redox process could occur. Plus, it's not readily available in stable forms suitable for widespread biological use.
Too reactive, it would just burn everything up.
Pretty much. So while theoretically possible for other biochemistries to support complexity, current scientific understanding still leans towards strong yet controllable oxidizers like oxygen being a critical ingredient for the evolution of highly complex, intelligent life as we understand it. It's those powerful yet gentle enough oxidizing agent available at planetary scales.
The Goldilocks oxidant, maybe?
We've explored oxygen as a life giver and a slow poison, a force that shaped our planet and our very biology. But what if we could fundamentally rewrite that story? Let's now explore the frontier of synthetic biology and bioengineering, where scientists are working to design novel, oxygen-independent metabolic systems, enabling the astonishing potential for human adaptation to extreme extraterrestrial environments and the radical conceptualization of post-biological existence.
Yeah, this is where things get really speculative but incredibly fascinating. Let's try to imagine a scenario maybe 30 years from now.
Okay, future gazing.
A young bioengineer named Lena is a prime candidate for the Europa Clipper follow-on mission, a mission to establish a long-term research outpost beneath the moon's icy shell.
Under the ice of Europa. Wow.
But she isn't just training, she's transforming. Lena has volunteered for a new class of gene therapies and synthetic organ integration designed to create a hybrid metabolic system, one that doesn't rely on the volatile, destructive cycle of oxygen.
Engineering herself not to need oxygen.
Exactly. Her body is being re-engineered to thrive on chemosynthesis in the dark, pressurized environment of a subsurface ocean. I imagine her talking to her therapist not about technical fears, but about the philosophical ones. She describes the recurring dream of forgetting the smell of cut grass after a rainstorm, or the simple pleasure of taking a deep, clean breath on a mountaintop.
Losing that connection to Earth.
She is choosing to leave behind the biological trap of oxygen, the aging, the cancer risk, the cellular decay, for a chance at a different kind of existence. But in doing so, she's severing a connection to Earth, and perhaps to a fundamental part of what has defined humanity for millennia.
That's heavy.
To me, this imagined story isn't just science fiction. It's the ultimate expression of the article's core idea. If oxygen is a double-edged sword, what does it mean to be willing to put that sword down entirely? Lena's choice represents humanity's potential future. To engineer ourselves beyond the limitations that nature imposed on us. It's an incredible, awe-inspiring prospect, but it's also deeply melancholic. It forces us to ask whether our destiny is to transcend our biological origins, even if it means leaving a part of our humanity behind on Earth.
Transcending biology itself. That's a profound thought experiment. So what does this imagined future mean for our present understanding? This idea of life without breath is no longer just fantasy. It's a serious area of scientific inquiry. It makes us reiterate the concept that oxygen, while absolutely essential for our existence, truly became a biochemical drug, an elegant trap evolution designed.
The golden handcuffs of metabolism.
Right. Life was forced into a profound trade-off. Immense power for energy, but at the cost of accelerated aging, sacrificing longevity for metabolic speed, and introducing the constant threat of cellular erosion. But the remarkable thing is, Earth itself holds the blueprint for life without oxygen in the form of anaerobic extremophiles.
The ones that never took the oxygen bait?
Exactly. These organisms are truly living fossils, thriving today in environments utterly devoid of oxygen, places like deep-sea hydrothermal vents, acidic caves, or boiling mud pools. Instead of oxygen, they utilize alternative molecules like sulfur, hydrogen, or iron to power their life processes. They literally remind us that oxygen isn't inherently life itself, but rather a dominant habit that evolved on Earth due to the unique circumstances of the Great Oxidation Event.
Proof of concept from nature itself.
They are living proof that biology is far more versatile than our oxygen-centric view often suggests. Now, in cutting-edge labs at places like MIT, Caltech, and NASA, scientists are actively working to rewrite life's blueprint through synthetic biology and bioengineering.
Building Life 2.0.
They're no longer just observing. They're trying to design entirely new metabolic systems that are oxygen independent from the ground up. This research spans various areas, including the development of artificial organelles, essentially, tiny engineered internal power plants that don't need oxygen.
Cellular power packs.
They're engineering entirely new oxygen-free respiration pathways, looking for alternative chemical reactions that can generate energy without the corrosive side effects of oxygen. This kind of work is critically focused on creating cells specifically designed to survive and thrive in extreme extraterrestrial environments like the subsurface, dark, high-pressure ocean of Europa or the thin, reactive atmosphere of Mars.
Designing life for other worlds.
This revolutionary work even opens the door to the conceptualization of post-biological humans, beings who could extract energy from internal bioreactors, making lungs and Earth's oxygen-rich atmosphere entirely obsolete. Wow, internal bioreactors. This incredible prospect means humanity might no longer be bound to a single fragile planet, but it also raises profound questions about what it means to be human if we shed our most fundamental biological processes.
Huge philosophical questions there.
This research into life without breath also offers critical insights into understanding hypoxia, the condition of oxygen deprivation, with vast implications for both human health here on Earth and for the demanding challenges of long-duration space travel.
Hypoxia, low oxygen, very relevant.
For instance, the groundbreaking research of David Ho and his colleagues revealed astonishing new ways cells survive oxygen deprivation. They found that cells can dynamically reconfigure their protein output, a process they termed translatome remodeling, as a powerful strategy to adapt to changing environments.
Translatome remodeling, okay.
Instead of just changing which genetic recipes, mRNA, they read, cells can selectively prioritize which proteins to actually produce and how many copies, while suppressing others.
It's not just reading the blueprint, but deciding what to build and how much.
Exactly. This fundamentally challenges the traditional transcription-centric view of gene expression, showing that the final assembly line for proteins is far more responsive and intelligent than we previously understood. This research also led to the discovery of a completely new mechanism, EIF4FH, an alternative translation initiation factor that gets activated specifically in hypoxic cells.
EIF4FH, a new player.
This fundamentally changes our understanding of cap-dependent translation and how protein synthesis is controlled, revealing a powerful new switch for cellular adaptation. Furthermore, from an evolutionary perspective, it's becoming clear that evolution seems to place stronger constraints on protein expression and translational dynamics than on simple mRNA levels.
So controlling the protein production is more tightly controlled by evolution.
It suggests that, these protein-making processes seem to be more conserved across species, and that translation plays a far more prominent, fine-tuned role in regulating protein expression than previously thought, making it a master controller of cellular function.
Fascinating. More control at the protein level.
From a drug discovery perspective, Ho's work reveals a unique window of therapeutic opportunity.
Practical applications.
By specifically targeting factors involved in this hypoxic protein synthesis, it might be possible to eliminate hypoxic cancer cells, which are often notoriously resistant to conventional chemo and radiotherapy, while minimizing collateral damage to healthy, oxygen-rich cells.
Targeting the low oxygen cancer cells. That's clever.
This understanding also has significant implications for the development of cutting-edge mRNA-based therapeutics, helping researchers better assess the translatability of potential mRNA drugs, meaning how effectively the mRNA will actually be turned into functional proteins.
Making sure the mRNA drugs actually work as intended.
Right. And from a broader perspective, it supports the growing consensus for multi-target therapeutics, particularly for complex diseases like cancer, by providing a deeper understanding of cellular signaling networks and expanding the range of potential drug targets, opening up entirely new avenues for treatment.
More targets, better treatments, hopefully.
We've journeyed from the rust on an old car to the very fabric of our cells, from Earth's ancient toxic atmosphere to the imagined future of humanity beyond breath. It's a profound story about a molecule we often take for granted, a molecule that is truly a paradox.
It highlights that life, as we know it, is a delicate and dynamic balance. The immense energy that oxygen provides comes at a cost, a constant microscopic battle against decay. But our ever-deepening understanding of this paradox, coupled with our boundless ingenuity, means we are no longer passive participants in this process.
It's a striking truth. We don't just age and decay because we stop breathing. We age and decay precisely because we never stop breathing.
That's quite a thought.
But that truth, rather than being grim, becomes an incredible testament to life's fundamental adaptability and human ambition. We are thinkers, makers, hackers of evolution, always pushing against the limits nature imposed.
It's a powerful reminder that what we call living is a dynamic, ongoing negotiation with fundamental forces, from the tiniest molecular interactions to planetary-scale transformations. And our future is increasingly about whether we choose to transcend those biological origins and redefine what it means to exist.
So breathe deep. Appreciate the profound paradox in every inhale and exhale, and consider how far we've come and how far we might still go in rewriting the story of life itself.